technical glitch we are having out here. So the meeting link was not working. So that's why it kind of took a little more extra time, more than 10 minutes, something like that. So I think all of you are here. I can see almost 45 participants out here. So again, the same thing, that please mute your microphone while the things are going on. But you can always write your question in the chat. It will be answered, and also you can raise your hand and ask questions accordingly. So today is the second day of the Introduction to Bioinformatics lecture course, which will be spanning for 3 months here. And the last day we were kind of having an ice-breaking session to make you understand about the how to log on into the educational site, how to log on into the server. Most of you have a login and a password to the server, and many of you, I guess, most of them, I guess, are having problem in logging on to the server itself. So the password seems not to be working. So we have kind of identified the issue. So Apar Agrawal is in charge of that. He has kind of worked very hard to kind of understand what the issue was out there. He has so Apar, you are there. Can you hear me?
Yes, sir.
Can you take the stage and tell them about the details of it?
Sure. So hi everyone. We know that you have been facing issues with the login details of the server account, but sorry for that, and we'll be up and working with the server details and the credentials. The credentials will remain same, and you will be able to log in with those same credentials to the server platform latest by tomorrow. So have a little more patience, and sorry for the inconvenience to all of you. Okay, sir. Thank you, sir.
Yeah, thanks, Apar, for the information. So I think most of you have been able to log on into the educational site as well and look around the projects and all these things. If not, I would request you to do it as we go on. So today is the second day. Before delving deep into the kind of things that you can right now see on the screen. And one more thing I would like to tell you, that whenever we send emails about the meeting links, etc. So if you're using Gmail, it will be in your promotion tab typically. So check out in the promotional tab. It will never be in the primary tab. There are several tabs in Gmail. So primary, update, promotion, and some other thing also. So primarily it will be in the promotion tab. Check the mails out there. We send out emails in time. So before raising a hue and cry over not receiving the email in time, please check out your promotion tab before, and we'll find the meeting link out there accordingly. So moving on, again, the same thing, a little bit of review since the last time we had the class. So this is the organization page. You can see the link right below. Please go on to the organization page to find out about the curriculum, to find out about the dates. Many of you ask questions that when is the second session. Today itself asked in the WhatsApp group that when is the next session going on. And please be very sure about it, that all these things are kind of specifically mentioned here in this particular website. So if you go to the curriculum, you will understand all the things that are being taught, will be taught, is being taught, all the dates, the timelines, the time, the things that will be covered, everything will be there. And I have, there's a forum out here where you can find many, links which we post regularly, or, as I was telling you, we are trying to create a very active bioinformatics community by virtue of this program. And also we have similar, upcoming programs coming up where the main target, the main, motto of this workshops is to give you that understanding about bioinformatics and have positive discussions and interesting discussions about the things that are going on around the world to kind of enable you and usher you into this domain of bioinformatics, which is gradually becoming a big importance nowadays in modern biology. And with the advent of data science and with the advent of the statistical principles which are used nowadays in the era of high throughput data generation, these forums are important to learn, because learning is not kind of segregated or specifically in books or specifically in the textbooks, etc. There are thousands and thousands of URLs, thousands and thousands of blogs and websites which actually shell out a lot of information on this big data bioinformatics. Bioinformatics as a whole, bioinformatics has gone through a paradigm change nowadays for the last 2-3 years, and it is kind of offering you all those exquisite technical things. Even if you are a non-technical person coming from a domain of biology, you need to kind of understand that to keep yourself afloat in this domain. And but be very sure that we are here to help you. The whole and sole purpose of this program is to really upskill you people, to really enable you people, even if you are a postgraduate, even if you are a you are just a simple graduate, or let's say you are a PhD or a postdoc. You are kind of, you will have equal ease in going through the resources that we're going to share with you, or we're going to tell you in these programs across. And one more thing I would tell you, that this is a class where we kind of discuss things, we kind of address the pointers. We'll definitely give you, we will be giving you pointers to the study. So you have to go back to the educational side and do some courses out there. That's important. The course is actually nothing but your textbook. And furthermore, there will be thousands and thousands of things out here, and we'll be posting some interesting links in the forum. You can go out there and check it out, check those links out. And this is the beauty of e-learning, so that you get access to those things even as long as you have a decent internet connection. I mean, most of you have, I guess, because most of you will be using a lot of Facebook and LinkedIn and all those social media platforms. Going forward, this is the progress that we have seen. It's there in the educational side. So as you can see, that there is an education part and there is also a server part. So Shubhrajit happens to be one of our research interns. He's also there in this meeting. Shubhrajit has been working with us for a long time. He uses a lot of server pipelines to analyze breast cancer cell line data. So that is why his server points of kind of is more than any one of you. But be very sure and do not panic. You can also do the same thing. So once you learn about that stuff, once you go along with us, we can show you, and you will be also definitely using the server platform. So I know that server is an issue right now, but it will be solved very soon, as Apart said, by tomorrow latest. You can get access to the server. But again, getting into the server is not the only thing. You have to understand what sort of input data, what sort of output data will be coming out, and how to analyze and how to get an insight out of it. What is the biological insight that you kind of have to, you can expect from the server itself. Among the rest, Ayushman, Devangana, and is there. You can also see your favorite teacher, Chitobroto, who also use a lot of server because he is also into part of it. And I would request you, Paul Joy, if you can kind of mention your full name out here so that we know you by the name, and also each of us can identify each other in a proper way. Satyanarayan Narayana is a person who has uploaded a photo in the educational platform. So I would request you to do the same thing. So going forward, this is a good profile. See, Ayushman has uploaded a good photo of himself, which he looks very smart and nice. Devangana is yet to upload a photo like that. So the rest of you, please follow the footsteps of Ayushman and Satyanarayan, so that we kind of see you. I mean, all of us who are there in Pine Biotech right now have photos out there, which is important because this is also a forum. So we know you by the face, we know you by the name, your good name, and also your achievements. You can share your achievements across the board. You can share your achievements in all the social media and so on. So as you go on doing the educational courses, as you go on doing the server, running the pipelines in the server, you will be kind of getting into the depth of it. Yeah, so going forward, so this is the first slide today about the data-driven hypothesis, data-driven discovery, and hypothesis-driven discovery in biology. So as you can see many boxes, right? You can see design a model, analyze the data, identify the question, review findings, gather data, implement, etc. Now, these are all building blocks. These are all building blocks in kind of trying to understand a particular biological problem. When it comes to hypothesis-driven data analysis or data hypothesis-driven discovery in biology, it all starts with an observation in the laboratory or a clinic. And we try to kind of get down deeper into that, trying to solve that, form that into a biological question. And we all use these building blocks, connect them semantically or meaningfully together to really have a hypothesis-driven discovery. The same contents, what you can see right now, all these blocks are also kind of reoriented and attached in another interesting way to come up with a different type of questions when it comes to data-driven discovery. So data-driven discovery totally depends upon the data, and hypothesis-driven discovery, which we have been doing for this time, for all this time, since a lot of time, because many of the scientists, most of the scientists who study biology, who study many of the scientific disciplines, they are kind of into the thing of understanding or looking at an observation and then trying to break it up and trying to do the analysis. Things will be a little clearer as we go on. So what is a biologist's perspective? So as you can see, he's a smart man with a pair of glass and a lot of retorts, and of course there's a computer in front of him. Do not mind the arrows out here. It is something that will disappear. So here it is. So what is a biologist's perspective? So a biologist's perspective starts with an observation, as I was telling you. So while doing an observation can be in a clinic, observation can be in a laboratory, observation can be in the nature itself. So from there, he or she kind of frames a biological question. So from the biological question, the biologist tries to kind of frame an hypothesis. Okay, whatever we see, whatever observation that we can see is probably for a specific reason, which might be XYZ. Now, to really understand the reason behind an hypothesis or reason behind the observation that he is making, he does a lot of experiments. So a lot of experiments which is connected with which is typically called data generation, and it is also definitely collection of the data. And thereafter goes into the analysis of the data. Now, what do we mean by data analysis? Data analysis typically means that you can use certain, you can gather all your data and come back in your research bench and try to connect the reasons behind it, try to connect the results semantically or meaningfully according to your knowledge. So that is why we call it knowledge driven or hypothesis driven. So everything is hovering around a hypothesis. So whatever hypothesis we are trying to we are framing, the data generation will be connected to the hypothesis, and definitely the data analysis will also be in line with the hypothesis that we are trying to do. And at the end of this exercise, we either support the hypothesis, we say, okay, this is true. Okay, whatever hypothesis that we have drawn from the observation is kind of true. And definitely, if it is not true, or if it is not really supporting, or if it is not really, not very congenial, or finding some negative results, I will refute the hypothesis and therefore report the results. So a scientist would be going forward, a scientist with a biological perspective would be going forward in this workflow to understand about a scientific question. What happens what does a bioinformatician do? Bioinformatician is also a scientist. Bioinformaticians starts with the data. What data? What is that data? The data which we are generated by the huge number of experiments nowadays from different biological experiments across the globe. Specifically, we call it high throughput experiments. So we start with the data. The data has been generated for different purposes. Maybe the data has been generated for, understanding the somatic mutations in a specific type of cancer, or the presence of some type of bacteria which is having an outbreak in a specific continent. So this can be the data source. So it can be any data. So we start with the data. We analyze the data first. Now, after the data analysis, the results that we get out of the data, we try to frame an hypothesis. So as you can see, it all starts with the data. It does not start with the hypothesis. That is why we call this knowledge-driven or data-driven science. Now we frame the biological question, what we are trying to address. From the hypothesis itself, it's a kind of a back calculation, because you already have the data in your hand, and you do the substantial amount of data analysis and then come up with the hypothesis, and therefore frame the biological question. After framing the biological question, there is again a biologist who does the experimentation. The experimentation is again nothing but data generation. But this data generation and collection is more specific. It's not like this data generation. This is sort of, a very elaborate sort of experiments which are carried out for a substantial amount of time, which are carried out for a substantial amount of time, and therefore it goes on like that. Now, this data generation and collection is something like which is very specific. Now, if might be so that if I am kind of finding a biological question, let's say I'm trying to identify a specific type of mutation in a specific type of disease or in a specific type of organism. Now, the experiments, the data generation experiments mentioned out here will be only around that. So I'll be kind of predicting this part that, okay, this is a these are the specific mutation you should look on. So thereafter, in the experiments, we create those PDX models, those knockout mice models in the biotechnology lab or in the science lab or the biology lab, and try to see the activity of those specific features. We try to identify those specific. So this is like a, we in this situation, it is like a needle in a haystack situation. We do not know where the needle exists. Bioinformatics help you to really understand where the needle is, or the area in which the needle is. So thereby we do some specific experiments, and thereafter the same process follows, like supporting the hypothesis or refuting the hypothesis. So from all these exercises, all these data generation exercises, what happens? It happens like a lot of data is being generated. A huge amount of data from a large number of tissues, from a large number of samples, from large number of cells, from large number of organisms. So if you really kind of think about the number of organisms that exist right now on the face of the earth, and also the number of tissues that each of these organisms have, and the types of cells that these organisms have, or the specific type of cells in these tissues, you can easily imagine the huge deluge of data that is there, accumulated somewhere. Itself, in National Center for Biotechnology Information, NCBI, which is a data warehouse, where lots and lots of data are being collected and are being deposited, are being maintained out there. So itself, in the gene expression omnibus repository, which is, the DNA and the RNA expression that are being generated from the high throughput experiments, only gene expression omnibus repository have almost 80,985 public data sets, hundreds of tissue types and thousands of organisms. So this forms the basis, this forms the source of our data for us who are kind of aspiring bioinformaticians out here. And we also can take the advantage of this data and kind of frame, do the necessary analysis, and also can do, derive hypothesis out of it. So at the end of this exercise, this course, when we come to the point that we are trying to do a project out here, these data sources will be used. These data sources you can, leverage upon. These data sources you can always get back to really frame that biological question and do the necessary bioinformatics analysis. Okay, so moving on. So all this data which is being generated nowadays, we call it big data. Sarah, could you mute your microphone, please? Thank you very much. Okay, so we call it big data. So why do we call it big data? We call it big data for 5 specific reasons. Which has it has volume, it has a huge amount, because terabytes and exabytes of data of our data being produced from the high throughput experiments. Huge amount of data is being produced. So that's why it has obviously has volume. Now it has a velocity. What do you mean by velocity? Now velocity is that the generation of the data is the rate of generation of the data is very, 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 very high because of the cheapness, the high throughput technologies or the scientific technologies that are being kind of advancing every day. The rate of generation of this data is very high. That's what we call velocity. So we call it data in motion. Okay, from milliseconds data to microseconds data, and you have to respond. I mean, in biological cases, maybe the response is not that very high, but when it comes to critical cases, like a person suffering from some type of terminal cancer, maybe the amount of data that are being generated from that particular from that particular person should be analyzed is very fast to come up with a plausible therapeutic procedure or a therapeutic intervention. But in other cases, there are thousands and thousands of mobile phones are now used. So communications, the chiefly in the domain of communication, these milliseconds and microseconds matter most. The amount of data that are being generated is huge out here. That's why we call it velocity. This has variety. Obviously, it has variety, and this is mostly unstructured. We do not know. There is no structure to it. So the huge job, the major job of a bioinformatician when it comes to really trying to understand if you're working with biology, if you're working with some other scientific disciplines, you need to have that basic knowledge about that. If you have to kind of have that basic understanding about the data types, because if you don't have this, you cannot really structure the data and come up with a process to make it or analyze it in such a way which will give you a meaningful insight. And it has veracity. And because there are since the purpose of the generation of the data, we do not know the purpose because somebody has done it only trying to understand all the somatic mutations of a particular cancer, or maybe somebody has kind of sequenced all the genomes that are available in the soil outside his lab just for his research interest. But we can use that. So we do not know the purpose of the data generation. That's what we call veracity. So there might be incomplete. So some situations might exist that you might have to literally throw away the data totally. You don't need to use it at all. You cannot use it at all. So that's why you have to understand what makes a data complete, or what are the basic principles of trying to look at the data, and what sort of analysis you need to do on the data itself to understand that how much sense the data would make. This is also a job of a bioinformatician, to trying to understand that how much sense that is make, because if it doesn't make sense, you cannot frame that biological question, you cannot come to hypothesis, or thereafter the whole process actually falls apart. And of course, if you really try to kind of bind all these, solve all these problems like volume, velocity, and variety and veracity, if you analyze it in a in a proper way, in a sane way, in a way which is kind of aligned with your interest or aligned with your biological question, it will of course add some amount of value to the entire process. So we know, we kind of say that this data, we call it 5 V's. We call it the 5 V's of the big data. The next slide is somewhat kind of maybe a little bit confusing, which say that it has 10 V's. Now, this particular slide is specifically taken from a publication, which is mentioned right here. So it says that when it comes to, this is a very biological case, to be very specific, If you see on the left, this talks about WES data. Now, what is WES? WES is whole exome. Now, people who are studying biology for the first time, or maybe in the undergrad days, maybe you don't have much access or much awareness of this particular word. But people who are kind of postgraduates out here, and also there are scientists I know, they know a lot of this whole exome thing. So in our eukaryotic genome, there are two sections of a gene which we call introns and exons. So exons are the part of that DNA which actually get translated or actually get transcribed into the RNA. So the RNA might produce some, some meaningful molecules in the form of proteins, which we call by mRNA. But there are other sets of RNAs as well. So the exome is that part of the DNA which actually makes those protein imprints, or produce that functional protein in the body. So this particular big data big data picture that you see out here is connected to the exome sequencing. So you can see that exome, there are, an exome is like every 5 minutes, this amount of exome are being produced in the body. These are being translated into, coming into the, in the cell level. If you really segregate that amount of thing and try to sequence it and try to kind of use the experimental procedures to decode them, you can easily imagine the amount of rate in which the data will be produced. And at the same time, obviously volume, BAM is a format. BAM is an NGS format out here, which we will address this as we go on. If you have this slide with you, the reason why I kind of gave this slide to you is not to confuse you at all, but to really give you a feel about the bigness of the data, the immense volume of the data, the immense V's that each of these big data contain. So, and most of the limitation of this, for this data is basically the hardware and the software limitation, and thereby, that is the whole reason we have this platform, we have this server, where where you can kind of log on, and definitely will be able to use that platform to visualize, because it's a huge data. It's not impossible for a simple configuration computer that you have right now in front of them, or even I'm using the computer that I'm using right now in front of me. It's not sufficient. The RAM and all these things, the memory and the hard disk and all those visualization specific data cards and all those specific the graphics card, etc., will not be able to handle it. And furthermore, it requires something very big. It requires something, a lot of processing is needed, a lot of back-end processing. To even load a 1 terabyte file in a simple computer, you can try it probably, but maybe it will, your computer will just say that, okay, thank you very much. I mean, thank you for your service, and I'm kind of quitting right away. So that's why you need some server. That's why you need some amount of, some diligent computation, some big resources, and computational resources, to be very specific, hardware and software resources at the same time, to really visualize it. And now obviously, the volatility is important. Sometimes we will come into this later on, and definitely, whole exome has some vulnerability. Vulnerability is connected with privacy and security, because somebody who is kind of maybe have some very specific disease which he or she doesn't want to reveal to the world always would ask for data security. So data security is also a very big thing, which is important when it comes to big data. By virtue of being there on the internet, you can access almost every sort of data around the globe. But the point is that whether you have the right to use it or not, because there are a lot of copyright infringement available. I know copyright infringement happening, which might make you land up in a lot of trouble. So that's why this part is very important. Okay, so and veracity and variety, we all talked about it a few minutes back. So keep this slide in mind. We will go into this later on.
Pratik,
yes, Mohit.
Srijita has a confusion in veracity, and she's been asking for you to, if you can explain it once more, the veracity part again, please.
Okay, sure.
Thank you.
Yep. So Srijita, So, when it comes to veracity, we all talked about the veracity specifically deals with the uncertainty or the data inconsistency. Because what we mean by the data inconsistencies, the data that you are handling might be incomplete. It might not be very useful for you to really do the analysis that you are kind of planning to do. So before using that data, you have to understand whether the data is complete by itself, or maybe whether it is valid at all, whether it is at all kind of useful for you. So that usefulness is the other word for veracity. Did you? I think does it answer your question, Srijita? Okay, you can write in the chat. Let me see the chat. Okay, thank you. Yeah, thanks. So going on. So as a result of this huge amount of experiments out there, we have several levels of cellular and subcellular structures. It all starts with the genome and gradually moves on to the transcriptome, and thereafter the proteome and metabolome and the epigenome. Now, what all these mean? Genome is the entire gene pool that are there right now inside your body. A specific part of the genome is actually transcribed into the RNA. The entire set of RNA that are being produced in the body is basically called transcriptome. That a little part of, as I was telling you, a specific, only mRNA really translates into the protein. So a specific part of a transcriptome actually get translated into the protein, which we call proteome. So the entire protein of your own body is basically of an organism, of the body of an organism is actually comprises the proteome. Now, as some, proteins have various functions. It can be structural protein. It can be, so it can be structural proteins. It can be some signal mechanism. It can participate in signal mechanisms. It can be hormones. It can be enzymes. It can be, a part of some cellular signals. It can be a part of your cell wall. It can be a part of your ribosomes. It can be a part of your, the histones, which are kind of very integral structural proteins of the DNA. So there are different types of proteins, but the proteins which take part in metabolism are the other proteins which play a very pivotal role to produce the desired effects or produce the desired effects inside your body. So the entire metabolic reactions. So metabolic reaction, by metabolic reaction, what I want to mean is that it's not about the digesting the food. There are thousands and thousands of, significant amount of biological reactions occurring inside your body to kind of communicate. Let's say right now you're listening to me, and maybe, the signals are flowing through your nerve cells to your brain. And these are basically reactions, and there are proteins which are participating into that. There are receptors, there are neurotransmitters, which are nothing but proteins. So they also play a role. So all these things are basically metabolome. So the entire set of proteins in action is called metabolome. Now, what is epigenome? Epigenome is something which is beyond the. Now this is a very, very interesting concept that has recently come up in the biological world. We used to know about this EPM, that part of DNA which might be, translated or transcribed into RNA, or might not be transcribed into RNA. So epigenome is that part of the DNA which is most often covered or masked by the histone proteins that we have in the DNA. And most of the effects nowadays, as we are gradually understanding the biological principles in a much better way, we can understand that many of the effects that we see, the difference between two individuals, the difference between two disease, difference between the same disease between two individuals, are largely controlled by the epigenome. One more thing I have kind of deliberately missed out from here, which is called metagenome. Metagenome is something which are those organisms which are basically single cellular organisms which lives in the soil, or they live in some extreme climates like hot springs, or let's say sulfur streams, or let's say very cold places like Antarctica or Arctic, something like that. So that is a different aspect. But metagenome obviously plays a part in our body, because metagenome is something which you have in your body. We'll come to it later on. So this is the flow. So from the genome to the transcriptome to the proteome to the metabolome, and of course epigenome. So epigenome is basically have bits and pieces of effect upon all these other factors out here. So where did the big data come into existence? So it all came into existence from the Human Genome Project, which was envisaged in the year of 1996, probably. And so the HGP took 13 years and a whooping amount of $3 billion US dollars to sequence the first human genome. Now, nowadays, due to the advent of these, very interesting, very high throughput technologies, very advanced technologies nowadays, we can even sequence our genome for $1,000, $1,000. So that's why there's a database which we call 1,000 Genome, and these gradually gave rise to the big data, large amount of large volume of information, and obviously collecting them, and thereby, as always, the researchers from different backgrounds, researchers from different fields of study have come together, have joined the efforts to analyze, to understand how does the human genome look like, and how does this human genome function. It was an understanding before the advent of this human genome. So when people were kind of sequencing a lot of bacteria, a lot of lower order organisms. Now typically Escherichia coli, E. coli have 60,000 genes, 60,000 genes in their body, which are into action. Now Escherichia coli is comparatively a very simple organism. It lives in a very simple way. It's a single cellular organism, so it kind of does not have any complicated metabolic process inside its body. So people thought that humans who have so much amount of complexity, and also at the top order of the phylogenetic cycle or phylogenetic ladder, the most advanced creation right now on the face of the earth, it might be having more than 60,000. It can be maybe in lakhs. But when it came to actually analyzing the genes, analyzing the genome of human genome, we came to see that we only have 20,000 genes in our body. Now, this was a real shock for the scientists. So that's why, it kind of inculcated a lot of interest among the people to look deeper into the stuff, that why is it so? Because we do not know. We do not kind of have some we know, that was the whole and sole reason why people started kind of with shovels and sticks. They came over to understand that what is happening out here. Why did our hypothesis go wrong? So that was the whole interest that kind of generated out there. So bioinformatics out here, as I was telling you, that it was kind of a lot of data were being gathered. So bioinformatics touched the domains of agriculture, where we try to do a lot of genome analysis of those specific plants which we are interested in, which we are kind of growing every day to produce our food, to have more production of the food, so that the world can be fed, the world can be supplied with sufficient food in the onset of increasing population. So thereby, we have to understand how the plant genomes work. So in MIT University itself, there's a professor out there who is trying to understand the, how does the tomato ripening occurs. So Solanaceae family among the plants is, if you notice, most of the vegetables that we eat every day belongs to Solanaceae family. If you take examples, I can give you 3 examples right away: potato, tomato, and brinjal. all these things basically belong to Solanaceae family. And if you really look at your diet, your everyday diet, you have this every day, right? So that's why it is important to understand, okay, how can we produce more, or how can we really fight the pathogenic disease that are kind of attacking all these plants, and have a very sane amount of production at the end of the day. So thereby, the scientists use the sequencing platforms to come up with the data. And as you can see, short sequencing reads. These short sequencing reads is nothing but the advent of big data. So when we have this big data, we need bioinformatics. We need the bioinformatic tools. We need databases. We need analytical platforms or servers to kind of find out that, okay, what is going wrong, or rather trying to understand what is happening out there. If you don't know what actually happens out there, how can you say that what is going wrong inside? So first you have to understand the natural process. So when people actually study cancer biology, the first question that a cancer biologist asks is that, okay, what is the normal function of a cell? You know, if you look at a tumor cell, myriad amount of functions are happening. But how can you say that which function is a normal function and which one is not? So thereby you have to understand, okay, what is a cell cycle? How does the cell cycle work? And thereby you'll be able to identify where the cell cycle is going wrong. So coming back here, so, identification of sequence gene and gene expression patterns. So all these data you can find in the NCBI and also some specific plant-related databases. And obviously, and the genome mapping, which is important because if we have the genome, we can understand that how the organisms work. So I will take a pause a little bit. If you have any questions, you can unmute your microphone and ask me, maybe. Okay, let's see the chat here. Okay, Devani, what is the difference between two data generations, a data-driven and hypothesis? Okay, what exactly is high throughput data? Okay, Akash, sir, can you repeat the function of epigenome? Okay, sir, when there is a data stored, is it stored in the server? No. Okay, from Srijita. Okay, so I will take up from Srijita. Devani, it is, where is the data stored? The data is stored in databases. A database is somewhere, is a hard disk basically, but of huge volume. It can store terabytes and petabytes of data, but a single database is sometimes not sometimes not useful, sometimes is not enough. So that is why we have a collection of a lot of hard disks around. It is sitting somewhere. Maybe it can be in your home, or maybe it can be on the cloud somewhere, where the data is being stored. So typically the data is stored in the databases. Okay, and okay, so Suprajit, thank you very much for the answer. Repositories. So yes, we call these repositories databases. Now a collection of database, which we call it data warehouse. NCBI, EMBL, all these things, European Molecular Biology Laboratory, or National Council for Biotechnology Information. These are all data warehouses, which has a number of databases dedicated to genome, to metagenome, to epigenome, to proteome, to metabolome, and all this stuff. So there are several sections. So let's say for a specific type of organism, you have specific type of information. So that's why they have made databases like ClinVar, clinical variations that we see in organisms. They have specific databases for clinical trials. So that's why they have a different database for clinical trials. So that's how it is. Hope I have answered your question, Devani. Okay, so can you repeat the function of epigenome? So epigenome is something which are, which are expressed and which are sometimes not expressed. The expression of that, so epigenome exists. Epigenome is something more than the genome. So epigenome, which is not really accessible for the this does not necessarily fall under the exons and the introns of the DNA, because they might not be exposed to methylation. Now, methylation is a very important concept, which we will come in the next lecture, when we talk about the DNA and the RNA. So epigenomic functions plays a big role in producing that 1% difference that you have with some other person. Besides other factors too, epigenome also plays a big role. So the epigenome in your body is some amount of genome which are expressed. We do not know when it gets expressed, and we do not know what kind of effects it might produce when it gets expressed. Maybe you will respond to the drugs much better. Maybe we will be resistant to some drugs in a different way. Maybe the common drugs that are being given to you, or a common drug that is given to somebody else, you might have two different reactions totally. Okay, Devangana says that I hope you answered your question, Akash. So what exactly is high throughput
Yes,
data?
sir.
Thank you, Akash. So Devangana asks that what exactly is high throughput data? So high throughput data is something which are generated by high throughput experiments. These are all automated experiments. These are all, the amount of data which are being produced in high throughput experiments is huge. Now, if you really, type this keyword in a browser, in a search engine to find out that high throughput data, what is high throughput data? You can find a lot of definition out of it. Okay. So maybe that will help you to understand a little bit better about things. So high throughput data is produced by high throughput experiments. And what are high throughput experiments? High throughput experiments are those huge data generating algorithms or processes which are used every day to produce that huge amount of data from different sources. Okay, I have a question, Devani. What is the difference between two data generation? Yes or no? Tell me.
Can I ask a question?
Sure.
I would like to modify the question Devyani asked about which one is more efficient. I want to ask whether the data-driven approach can exist independent of the hypothesis-driven approach. That's my first question. And the second question is that for big data, you said there are 5 V's, velocity, etc., and veracity also. So my question is, can all these properties be quantified? And even if quantified, suppose for veracity, how is it quantified? And if quantified, is it absolute or varies according to perspective?
So what do you mean by a perspective?
Perspective means does it vary from a hypothesis to a hypothesis, or veracity is fixed for a fixed data set?
Okay, so answering your first question, what was your first question once again, Arno?
The first question is whether the data-driven approach can exist independent of the hypothesis-driven approach.
No, not at all. Because we are trying to reach an hypothesis by the data-driven research. Hypothesis, what is hypothesis? Hypothesis is a semi-decision, which we are trying to prove.
A proposition.
Obviously, proposition, yeah, or a prediction, let's say. So we are trying to understand that, okay, I think that, let's say right now, let's say the sun is burning upon a particular part of the globe at an angle of 45 degree at the rate of something. Okay, this is a hypothesis. For doing that hypothesis, you have the data. We do not know what data. So when you do a hypothesis driven research, you have this question upfront. But when you have a data in the beginning, we try to analyze the data and come up to this hypothesis. We do not know beforehand that, okay, only after analyzing the data, you can reach that hypothesis, which is, only the analysis will be able to tell you that hypothesis. You cannot go without the hypothesis at all, because hypothesis is something which is equivalent to a biological question. You have to ask a biological question to the data. Now, when you are asking that biological question, reframing that biological question is basically the hypothesis. You cannot do without hypothesis in any sort of research. But obviously, hypothesis-driven research, which means you start with a hypothesis and then gradually go on collecting the data. And the second part is that you start with the data and you go accordingly along with, go in a definitive way to understand, okay, this is the hypothesis that I'm trying to prove. Did it answer your question?
Yes, sir. Yes,
and
sir.
What was the second one? Sorry?
The second one is, how are the values, the 5 properties of big data that you mentioned out there, the velocity, and I don't exactly remember
Yeah,
the
yeah.
others.
Velocity, volume, veracity. So you
Volume.
question on
How are
veracity.
how are they quantified? Suppose for veracity, since veracity is the degree of uncertainty or the ambiguity of the data,
Okay.
how is it quantified for a data set?
Okay, so we have quality scores. We have certain p-values. We have certain statistical principles. So when it comes to veracity, when we say our data is inconsistent, or let's say data is not very useful, you have to probably look for the kind of quality scores that a particular algorithm is going to give you. So before embarking into the actual data analysis, you might like to have a data, you might like to see the data, and be very sure most of the times, in many of the cases, to really understand, to really solve the veracity problem, you need to have that biological insight, which is sometimes beyond the qualities of the statistical principles that are being used. Let's say you visualize a particular data. Let's say you're trying to visualize the data. If you want to visualize the data, your basic understanding will tell you whether this data is complete, whether the data is inconsistent, or because you know you have to kind of, let's say you're looking for some mutational hotspots. And when you are looking for some mutational hotspots, you are kind of have some knowledge. I mean, you obviously should have done some amount of research on the type of mutations that you are trying to see. Okay, so let's say you are trying to understand that thing from the data itself, but then when you actually see the data, you see that this is inconsistent in the way that, okay, maybe it's incomplete, maybe the process in which it is generated, this is not giving you the right starting material for your own experiment. So thereby you kind of understand, or let's say quantify in your word, to about the veracity aspect of the big data. Does it answer your question?
Does it change from one biological question to another, the degree of
Not really,
veracity?
not really, not really, not really. So veracity means, it is inherent to the data. Whether you are answering the biological questions or you are trying to frame that any biological question, it is inherent to the biological data that is there. So veracity as a perspective, it might change, but typically it does not.
Okay, thank you,
Is that
sir.
okay? Okay.
Thank you, sir.
Warno. So yes, Shubhrajit has answered a lot of question. Devani, Devani, data-driven. There's a question of efficiency. Oh God, a lot of lines. Sorry. So data-driven is more efficient than hypothesis. No, I mean there's no question of efficiency. Definitely, data-driven. Oh yeah, obviously, data-driven is more efficient because it takes less amount of time and it kind of reduces the situation of the needle in a haystack situation. Yeah, Suprajit, you are absolutely right. Have some pros and cons because you have to have those specific understanding about the data, and also have to do a lot of kind of quality checking experiments before going into proving the hypothesis. Thank you, Mohit, for answering the question. Sreyoshi, sir, you said E. coli has, whereas humans have 20,000 as per AGP analysis. I mean, what is the reason behind it? Is it related to C-value paradox or any others? Well, it is not related to C-value paradox right now. Maybe it is, yeah, that's the definition of it. So, I mean, when it was sequenced, it was found that human have 20,000 genes. The expectation, yes, a part of it. Yes, definitely that's a complexity of the trait of that organism. But uh, what I was trying to mean by showing you the difference between 60,000 genes and 20,000 genes of human is that that's the hypothesis. Okay, if you see really, that's a hypothesis. When you are sequencing an E. coli, you have a 60,000 genes out there. We kind of hypothesize, okay, humans are more complex. It should have more number of genes. But actually, it is not so. That's what we find out. That's what we found out there. So yes, it's somewhat related to C-value paradox, but obviously at the same time, what Apar said is also right. That's the complexity of the trait that each organism has. Okay, so moving on. I think I have, yes, full screen. Oh God, sorry. Give me a minute. Yes. So we have applications in agriculture. We understood a little bit about it. So we have applications in defense. Okay. So when we have the primary idea of bioinformatics in defense, we start with that many of the soldiers we go to fight in other countries die of infections. In Calcutta, there is an organization called NICET, National Institute of Cholera and Enteric Disease. Now this organization has been there since the time of the British. The British people have set it up, and it is there to really identify the typical disease that the soldiers are kind of suffering from since the time of the First World War. And NICET have that stock of all those pathogenic organisms that affected the British soldiers, and there was a kind of epidemic of these people. So NICET was set up for that, and still now NICET kind of does a lot of research in virology, in bacteriology. They also have a very frolicking bioinformatics department. Now, when it comes to defense, on the left you can see that so influenza. So there's a please go to this website and try to find out whether what does it actually mean, because many of the people who will are kind of affected because of these domesticated organisms like pigs and birds. Recently, a few years back, Calcutta was kind of reeling under the pressure of bird flu. So bird flu was an H1N1 virus. So an H1N1 virus can, virus being very very mutable, it can kind of affect humans a lot of way, can produce a lot of your respiratory disease. And recently in Calcutta, there was one high rise where a thing was reported that people were suffering from a lot of breathing problems. So people when the doctors when the people went to consult the doctor found out that in one of the apartments there's one guy, or there was one family who was kind of rearing a lot of pigeons and doves. Now pigeons and doves they have specific type of bacteria in their feathers, which was getting into the duct of those those AC vents and was affecting that specific group of people. So bacteria and virus are very very difficult to handle, very mutable, very they can adapt to any sort of environment, and therefore can affect us. And thereafter comes the point that to really understand that how they can be kind of eradicated, or rather than that, they can be mitigated. There's also something called DARPA project, DARPA, DARPA. So that is called Global Biodefense. And please go to this website. You will find you can find this. You can go to this website and find out some details about it. And thereafter, we come to the application of bioinformatics is kind of booming up in the domain of healthcare, which says that there's a case which is described out here. So what is NSCLC? NSCLC is non-small cell lung cancer. In non-small cell lung cancer, there are three important genes which are kind of mutated, which are called KRAS, EGFR, and ALK. Now, to really understand that what these two, as I was telling you before, actually going into the process of understanding where things have gone wrong, you have to understand what normal roles does these three genes play. As you can see, KRAS, which is a protein that is involved in regulating the cell division. So as you can clearly understand that if KRAS goes wrong, the cell division will be haywire, right? What does EGFR does? EGFR is the full form of EGFR is epidermal growth factor receptor. It's a protein. So it acts as an extracellular ligand. So it kind of produces that adhesion between the cells. So if it goes wrong, the cell will kind of not be able to be static in a particular place, and they'll kind of roam about in many places, and maybe causing that that losing that heterozygosity of the cells, and obviously metastasizing into different tissues. What does ALK do? ALK is a tyrosine kinase protein, and this is important in transmitting signals. So As you can see, three significant metabolic functions, starting from cell division to receptors, adhesion, and also transmitting signals. If these three cellular functions goes haywire, it would be difficult for a particular cell to maintain its sanity and go into a level of the tumor. It can become the mafia of the cell. So when we come to this, bioinformatic application in healthcare, we are trying. They always try to from the patient clinical data, from the patient samples, from the genomic data, from the transcriptomic data, from the RNA expression data, RNA-seq expression data. They try to understand what are the genes that have gone wrong, what are the genes that are really producing that truant effect within the cell itself. So as I was telling you, metagenome, very important part, very important, becoming very important nowadays, that there is a significant project which is going on which says American gut. So they are trying to understand the gut bacteria that are present in our body, because we say there's a recent thought that the type of character of a person is largely guided by the metagenome that is there within our body. If you do, if you really look at it, the entire food that are being digested in our body, in our stomach, and also in our small intestine, are largely controlled not only by the enzymes but the amount of bacteria that are there inside our body. That's why the concept of probiotic drink, the probiotic capsules, probiotic potions have come into existence, because people have understood antibiotic will only kill randomly all the bacteria. That's why all the doctors nowadays prescribe a lot of probiotic associated antibiotics, or they are kind of they say the patient should go on through a probiotic course after having the antibiotic to revive the friendly bacteria within your body. The same thing happens in environment as well. When you apply a lot of pesticides, all the organisms die, be it friendly, be it unfriendly, they all die together. But there are processes by which you can really revive that beneficial soil microbes. A lot of this part of agriculture is called sustainable agriculture. This part of agriculture has a lot of NGS. You need to understand what are those bacteria that live within the environment. So American Gut Project is dedicated to that, and there is also the genome, integrated microbial genomes and microbiomes database. These are all repositories which are there. They are kind of regularly updated. You can go out there and figure it out, what kind of data do exist out there, and they all use a lot of high throughput technologies to gather, collect, and curate the data. In cancer biology, there's a lot of resources out here. They use, as you can see, SRA. SRA is called Sequence Read Archive. This is a part of NCBI. Now, ncbi.nlm.nih.gov is the data warehouse. Under it, there is Sequence Read Archive. You can find what you can find here. You can find here all those sequence data from different technologies, generated by different technologies. As you can see, Roche 454. These are all next generation sequencing algorithms which are used and which are getting advanced day by day. Therefore, the rate in which the data is being produced are huge. That's why we say velocity, and obviously, veracity is also has to be taken into consideration before actually going into actual data analysis. So we'll kind of move a little fast because we are kind of out of time. So there's another thing called GTEx. This is important because GTEx is a collection of data, is a collection of samples which are taken from normal samples. How does a cell functions normally? What is the normal gene expression? Because if you do not know the normal gene expression of a particular organisms, how can you say where the gene expression has gone wrong? The other cancer repositories, cancer data repositories are, discover.repository.io, and also TCGA, the Cancer Genome Atlas. These are, they kind of have almost, they cover almost a number of, therapeutic area within the cancer, a number of variations within the cancer, and there are a huge number of, patient samples in this particular database, which are open for, and some of them, obviously not all, some of them are definitely open for analysis by people in the anywhere in the world. Now, all these things, whatever I told you, is comprehensively kind of covered. Now, again, I'm telling you, this is only the tip of the iceberg that I am telling you right now. Because as you know, if you do not understand the basics of it, now where the data is generated, how the data is generated, which are the data that are going to be used, if you do not have a broad overview of everything, you cannot delve deeper into the stuff. So please go to this particular website, this particular course we have. This is the reading material that you're going to have as a part of this lecture that we have done today. And please go out there and do the modules, go through the modules, answer the quiz, and I can guarantee you at the end of the day, you will have an interesting experience and will definitely be a better place to understand the questions that you're asking me right now. You can have a better understanding of. There are a lot of videos out there, a lot of YouTube videos out there. It will take you an hour or so to go through the entire length of the courses. This is the study material that you're going to, you should direct yourself in. And after you complete this thing, there's a tab called review out here. So please add your review. How did you like it? Maybe you liked it, maybe you did not. Now, if you don't like it, it's okay. It's perfectly all right not to like something, because your point, your comment, or your view is very important to us, because we also need to know what actually who are the people whom we are catering to. And obviously, this will be helpful as a feedback mechanism to us to understand that what do you actually want, and we will be able to improve our courses. So Apar will also be sharing a feedback form with all of you in your email. Please, I would request you across the board, be it students, be it scientists, be it faculties, or anybody out here, to fill up that survey and get back to us, because that would definitely help us to understand your needs. That will help us to understand your requirements, and we can address that accordingly. Now, all these things, what I said, connected to metagenomics, connected to epigenomics, connected to transcriptomics and all these things, defense and agriculture and all these things, we have projects. I have shown you this slide last time also. So during this entire length of the course, you are free to go into these specific projects and do it at your own purpose. Again, I'm telling you, the responsibility of understanding stuff largely depends upon you. It's not possible for us to really go to your place, physically, and kind of, shoo you around to do the courses. The things are there, the food is there on the plate. You have to kind of, make your move to take up the spoon and eat the food. So it's very difficult to, so please don't be shy. Please don't be, kind of say that, okay, can I access this later on? So during the entire length of this course, you would get the access to all these projects. So these projects are created from very important high impact journals, hovering over the topics that are being covered in our educational platform, and topics that will be covered in the entire length of this course. So as you can see, all the concepts that we talked about are covered in this course, and go there and try to do it, hover around that educational platform. And the next day, we are going to touch upon the very important concepts of molecular biology, like DNA code, transcription and translation, and also quantification techniques, which are used for understanding the molecular activity at the subcellular level. And we will talk about, DNA variants and RNA expression analysis, and what is actually next generation sequencing, and what is it, and how can it be used for understanding the different biological principles, the different biological insights that we are intending to see from different cases across the globe. Thank you very much. So if you have any questions, you can unmute your mic, or I can kind of go to the, we already have 6 questions out here, I guess. So yes, Mohit have answered most of them. Yes, so I think most of the questions of you have kind of been answered. So today we had a hiccup of kind of today we had a hiccup of understanding your study.